So a couple things here about exam 3. First of all, it will be in Blackboard. It'll be all multiple choice questions. It'll be worth 50 points, so not 100 points. That means we'll only have 16 questions and 2 extra credit points on that exam. So each question would be roughly 3 points, a little bit over 3 points per question. You'll have to log in via Zoom, so make sure you're there promptly 9:15. And you will need a password to access the test, so you will not be able to do it unless you are present at the Zoom session. And I'm intentionally mentioning this just for students who may be listening to the recordings who may not actually be here today. Your video must be on and your mic muted so that I can see you. Not necessarily hear you, but just make sure that you are kind of focusing on the test, and I can check in and make sure everything is okay. And if you have questions, you'll probably be able to communicate with me through the chat privately, right? So you can ask me questions privately through the chat. Other things I want to point out is that we're organizing a lab TA session. So there will be a few sessions available on the 22nd, the 23rd, and the 24th. They'll be in the evening from 6 to 8 p.m. One of our TAs, Lauren, will be there. She'll be answering questions. For those of you who may need help, I think there was a spot or somewhere in the phys labs that instructed you to seek help from a TA if you need it. And TAs will be there answering questions about phys lab as well as anatomy content in preparation for the practical, but also just to help you if you need any guidance with the remaining quizzes. I think they'll be due at the end of that week, so the 24th. So be sure to put this in your calendar, either the 22nd, the 23rd, or the 24th via Zoom session. And I'm gonna send out an announcement with instructions on how to get into this channel. So this is a specific channel that we have set up in Zoom for all of the TA sessions. And I'll put the instructions in an announcement so you can access that Zoom channel and tune in on either of these days for that review, okay? Make sure you go to these sessions, guys, because I'm intentionally organizing them. Because I cannot answer questions face-to-face, I haven't really seen a lot of students come to the reviews or office hours, and I don't know how to help you guys other than to kind of set up these types of opportunities. So let me know how I can help you. Let me know what would be helpful. And then definitely reach out and attend these sessions if you think that you need this type of help, either with labs or if you need help with the actual lecture content too, okay? All right. So that's all I have in the way of announcements. I am going to review some of the stuff we talked about from the autonomic nervous system, just because we did this quite a while ago. Let me just make sure there aren't any questions coming up here. Okay, no. Just because we did this the longest time ago, or this is the topic that we covered at first. So this is what I'll start the review with, just pointing out important things that you need to know. These are drawings that I typically do step by step, but because of the kind of complexity of the drawings, I'm just going to go over it here, and I will post these images for you in Blackboard, okay? So this is an overview of the nervous system that we're looking at here. And I've divided it in the way that the neurons differ. So we have 3 different neurons that we can see here communicating. 2 of them are coming from the autonomic nervous system, which is the involuntary branch of the peripheral nervous system. And then the one at the bottom is representing the somatic or the voluntary branch of the nervous system, right? And so we're going to use these 3 neurons to speak about the anatomical differences between the 2 divisions of the nervous system, and then some functional differences as well. We'll talk about the differences in the receptors and the differences in the neurotransmitters at different locations throughout these 2 neurons, these 3 neurons rather. So we'll start by looking at the autonomic nervous system. And first off, we know that the fibers here arise from a different part of the spinal cord. They're coming from the lateral horn, as we can see here. And then they make their way into this first neuron, which we call the preganglionic neuron. We also want to point out here that there are two neurons in this autonomic communication, whereas in the somatic communication, we've only got one neuron. Okay, that's an important distinction. Other differences we notice here are that the segments of the spinal cord from which these fibers arise are going to differ depending on if this is in the sympathetic system or the parasympathetic system. So in the sympathetic system, we've got fibers coming from T1 to L2. We describe this as being thoracolumbar. In the the parasympathetic system, our fibers are coming from our cranial nerves, 3, 7, 9, 10, and then our sacral nerves, S2, S3, S4. So different regions of the spinal cord, but still coming from the lateral horn in terms of the location in the cross section of the spinal cord. So those fibers are going to merge into this preganglionic neuron, and that is going to communicate with a postganglionic neuron, so a second neuron at an autonomic ganglia, right? Now the autonomic ganglia, again, are going to differ depending on whether we're in the sympathetic system or the parasympathetic system. Here we can see, first off, in the sympathetic system, the autonomic ganglia is much closer to the spinal cord, okay? In the parasympathetic system, it's going to be much further out and closer to the effector tissue or the neuroeffector junction. Another difference here is that we've got a shorter preganglionic neuron in the sympathetic and a longer postganglionic neuron, whereas in the parasympathetic, we've got this long preganglionic neuron and a much shorter postganglionic neuron. We can also speak about specific to the sympathetic system. There were 3 routes of communication. So one is this ganglia, and we mentioned that this connection, this communication, this autonomic ganglia was what formed the sympathetic chain. So the collection of all of these ganglia that are going down the sides of the spinal cord, together that formed what we call the sympathetic chain. But that was only one route of communication for the sympathetic nervous system. The second route of communication was synapsing onto an adrenal medulla chromaffin cell. And that chromaffin cell would in turn release 80% epinephrine and 20% norepinephrine and a very, very minimal amount of dopamine as well. And then the third route of communication was a collateral ganglia, which was also an autonomic ganglia, but not a part of the sympathetic chain, okay? So those are the 3 routes that we spoke about for the sympathetic communication specifically. In terms of the anatomy of the sympathetic nervous system, we also described the different fibers that make up this communication. So we spoke about white ramus fibers or myelinated fibers going from the spinal cord and comprising the preganglionic neuron, and then gray ramus fibers or non-myelinated fibers comprising the remaining connection to the postganglionic neuron. So we had this difference in the myelination that we described as being either white ramus from spinal cord into the preganglionic neuron, and then gray ramus from the ganglia into the postganglionic neuron. And so those are some unique properties that we saw for the sympathetic nervous system. As far as the parasympathetic nervous system, there was only one route of communication. It's going to synapse onto its ganglia, not a part of any chain, so there's no parasympathetic chain. There's no such structure in the parasympathetic side. And then from this ganglia, that second neuron, which is much shorter, is going to go right out to the effector tissues. Let's describe what some of the effector tissues are for the autonomic nervous system. Those are going to be our involuntary tissue. So smooth muscle, cardiac muscle, glandular tissue, and then in the case of the sympathetics, adipose tissue as well. And so those are going to be tissues that we don't have voluntary control over and that are under the influence of the parasympathetic and sympathetic system. Okay, let's now speak about the neurotransmitters and the receptors. So here we want to be clear, all preganglionic neurons, right? All of them, this sympathetic preganglionic, this parasympathetic preganglionic, always release acetylcholine. So there's only one neurotransmitter that is being stored in this, this axon terminal in a preganglionic neuron. It's always acetylcholine. As far as the postganglionic neuron, this is going to differ depending on where this neuron is. If it's in the sympathetic system, the neurotransmitter will always be norepinephrine or epinephrine, right? Those are binding to adrenergic receptors. In the parasympathetic side, it's going to be acetylcholine as well. And we talked about one small exception to this rule, which was sweat glands, which is not a big deal, but just I want you to be aware that there is an exception to this rule. So postganglionic neurons in the sympathetic side release norepinephrine, which binds to adrenergic receptors. And then postganglionic neurons in the parasympathetic release acetylcholine. These are binding to muscarinic receptors. Okay. On the postganglionic neuron, the receptors, let's speak about the receptors now. So because all preganglionic neurons release acetylcholine, all of our postganglionic neurons, be it in the sympathetic or parasympathetic, they will have nicotinic cholinergic receptors on their soma. So on the cell body, this acetylcholine will bind to a nicotinic type of cholinergic receptor. And we want to differentiate that from the muscarinics, right? The muscarinics are not at the ganglia. We only have nicotinic receptors here, which are fast receptors. Another way of saying that is that they are ionotropic receptors. And another way of saying that is that the receptors are also a channel, okay? So this is going to be a fast response, and that's the type of response we want here at the ganglia. Out here at the neuroeffector junction, we talked about some differences. So for one, we've got different neurotransmitters depending on where we are. The second difference here is the type of receptor that's located on these effector tissues. So in the sympathetic system, we have our adrenergic receptors, and I'll talk more about the difference between our different receptors and affinities and so on. But here we have alpha-1, alpha-2, beta-1, beta-2, or beta-3 receptors on the effector tissues that are binding this norepinephrine. In the parasympathetic system, we have cholinergic receptors, again, because we're releasing acetylcholine here, but there are different type of cholinergic receptor. They're going to be muscarinic cholinergic receptors. Okay, really important difference to make, or distinction to point out here, okay? I also want to talk about the general difference between these two junctions, so between the neuroeffector junction and the neuromuscular junction. We talked about two differences. One was the distance of the cleft. So the distance between this ending of the postganglionic neuron and the effector tissue, the distance here is greater than the distance that we see at the neuromuscular junction, or the distance here at a neuron to neuron synapse, okay? The second difference is that we've got varicosities. So these swellings that are shown here at the end of this nerve terminal in the postganglionic side in the autonomic nervous system, these were described as being varicosities, and these were synonymous with the axon terminal. This is where that vesicle, that neurotransmitter vesicle, is being stored, and that neurotransmitter will be released here at the neuroeffector junction. So those are two differences that we established between the neuroeffector junction and a neuron-to-neuron synapse, such as here at the ganglia, as well as the neuromuscular junction, which is in the somatic nervous system. Righty? And then to end off speaking about the autonomic nervous system, I'm going to talk about the general functions of both of these divisions. So we know that the sympathetic system controls our fight, flight, and freeze response. This is going to be under scenarios where we're under threat or some type of danger. This is going to be elicited by the quick flooding of the system that is especially created by the adrenal medulla chromaffin cell. So this second route of communication where the adrenal medulla chromaffin cells flood the system via this hormone, right? Remember, because it's entering the blood here, we're describing it as a hormone, and that will flood the system very quickly, which is the type of response that we need in the sympathetic system, right? That fight or flight or freeze response that can be brought about very, very quickly. The general function of the parasympathetic system is going to be just the opposite. It's going to be involved in resting, digesting, and then glandular secretions, excepting sweat glands, so not sweat glands. Our other glandular secretions, this are going to be our exocrine glands, our salivary glands, the liver, the pancreas. Those types of glandular secretions will be enhanced under the parasympathetic influence. And so we spoke about the comparative secrecy that's exhibited between these two divisions of the peripheral nervous system. So these two systems are working in balance in order for us to be at rest, with the parasympathetic system slightly more predominant, right? Remember, at rest, the parasympathetic system has a little bit more influence than the sympathetic system, but generally they have equal contribution to keeping us in a restful state. If we're going to be in a more excited state, the sympathetics take over and the parasympathetics must be toned down. If we're going to be in a less excited state, the parasympathetics are going to drastically take over and the sympathetics must also be toned down. And all of that is elicited, all of that is sort of instructed in the integrating system, the integrating centers, that are located within the brain, right? The medulla, the pons, the hypothalamus. And lastly here, as we look at the somatic nervous system, we pointed out some of the differences already. The fact that this is voluntary, so we do have control over this. This controls our skeletal muscles only. So the only affected tissue here are going to be skeletal muscles. The area of the spinal cord that these fibers arise from are the ventral horn. So not the lateral horn that we see here, the autonomic division, but the ventral horn is where these fibers arise from, and they are going to exit the spinal cord, merge to become a part of this motor neuron. So here we only have one neuron, it's called a motor neuron, and it forms a motor unit. Remember that one motor neuron innervates many muscle fibers, but one muscle fiber can only be innervated by a single motor neuron. And so this connection forms the neuromuscular junction, which has only one neurotransmitter, always acetylcholine, and only one type of receptor, always a nicotinic or ionotropic type of receptor, again, bringing about a fast response here at the neuromuscular junction. And I think that's all I'm going to review for the peripheral nervous system. Make sure you have an understanding of the events that takes place at these two junctions. In fact, I think I have a drawing that kind of goes over that. Here we go. So this side-by-side drawing is representing what happens at the neuromuscular junction and what happens at the neuroeffector junction, okay? One, we have a varicosity here. Two, we have the terminal bouton here. This is going to be of our motor neuron. This is going to be of our postganglionic neuron. A second difference is going to be the receptors that are located here. So in the neuroeffector junction, we have muscarinic receptors or adrenergic receptors. Remember, this can be either the sympathetic system or the parasympathetic system. And the neurotransmitter is also going to be changed depending on which division we're in as well. We talked about the different mechanisms, the different signal transduction pathways for the various adrenergic receptors. So make sure you understand which receptors use cyclic AMP, which receptors use IP3 and DAG, right? There can be either of these two second messengers involved depending on which adrenergic receptor is located at that tissue. As far as the neuromuscular junction, it's always going to be a nicotinic receptor. It's always going to be excitatory, and that is always going to bring about an end plate potential. That's what we described the excitation of the skeletal muscle. We describe that as being an end plate potential, right? Always excitatory. Make sure you understand the steps of this event. I'm not going to go through these in detail, but make sure you understand how we go from vesicles with this neurotransmitter, action potential coming along, exocytosis, and that whole pathway, okay? Which is fairly similar in between these two junctions with a few minor differences. And then the last thing I'm going to speak about here as far as the autonomic nervous system is this table. So I asked you all to create, as a part of your homework or creating a study tool, to organize a table that looks something like this, that indicated the receptors, what tissues they were found on, what neurotransmitter they preferred. So what type of affinity they had for norepinephrine versus epinephrine, the effect on their second messenger, and then the effect on the actual organ or tissue, the affected tissue in question, okay? And so we talked about alpha 1, alpha 2 being very similar in their locations, right? Most of our vascular smooth muscles where we find these two types of receptors, also similar in their affinities. Both of these preferred norepinephrine over epinephrine, but their second messengers were different. They had two entirely different signal transduction pathways. Alpha 1 receptors used IP3 and DAG as their second messenger. Alpha 2 receptors use cyclic AMP as their second messengers. Alpha 1 is going to be connected or coupled to a G stimulatory protein. Alpha 2 is coupled to a G inhibitory protein. And then the effect on the affected tissues are pretty much the same. These are always going to be excitatory to their affected organs or affected tissues. As far as the beta 2 receptors, we saw quite a bit of difference. Beta 1 receptors were on the heart and kidneys, beta 2 receptors on the respiratory tract and the uterus, so the smooth muscle that's found here in the respiratory tract and uterus. Beta 3 was mostly adipose tissue. Beta 1 and beta 3 had equal affinity, so norepinephrine and epinephrine bound with an equal affinity to these receptors. Beta 2, however, had a higher or increased affinity for epinephrine. As far as their second messenger, they all use the same second messenger, and they all were coupled to G stimulatory protein, so they're going to be enhancing or increasing the second messenger, whereas alpha 2 is going to be decreasing its second messenger. But the effect on the tissues were slightly different. So beta 1 and beta 3 were excitatory to the tissues, to the heart, kidneys, and adipose, whereas beta 2 is going to be inhibitory on the lungs and on the uterus. It's going to be inhibitory to these tissues. And I spoke generally about what excitatory versus inhibitory means. An example we talked about here with the lungs is that inhibiting this tissue creates relaxation of the smooth muscle that lines the airways, which allows more airflow into the lungs. And that is in alignment with the overall function of the sympathetic nervous system, right? Increased oxygenation by relaxing that smooth muscle on the airways and bringing more air in. And that is the type of inhibitory action that we're speaking about with beta 2 receptors. Okay, so make sure you have a clear understanding of what it's doing to the effector tissue and then what it's doing to its second messenger, right? Two entirely different effects. Okay, so I'll pause here to see if there are any questions that come out from that or anything else. I don't see anything just yet. And then in the meantime here, I'm going to pull up another drawing. Okay, this one. All right. Right, so I don't see anything coming up in the chat, so I'm assuming everything is okay so far. One of the other things that I wanted to kind of review was this table that we drew in class. So this is the hypothalamic anterior pituitary hormone axis. And this really just is a general overview of the hypothalamus and the tropic hormones that are released there, the anterior pituitary gland and the tropic hormones released here, and then the various endocrine glands that are being influenced, and then what they're doing, the hormones they're secreting or the effects that they're having as a response to these trophic hormones from the anterior pituitary. So this axis that we described, so make sure you understand the effects of all of these hormones. So which trophic hormone from the hypothalamus stimulates which anterior pituitary hormone, whether it's stimulating or inhibiting, whether it's a releasing hormone or an inhibiting hormone, and then the action that is brought about by the endocrine gland itself. We spoke a little bit more in detail about this axis, about the thyroid hormone axis. So make sure you have an understanding of what thyroid hormone does, right? Its action on basically the metabolic activity of cells. Make sure you have an understanding of the difference between T3 and T4. Right, T4 is more involved in the negative feedback loop. T3 is more involved in being active or bioavailable to tissues such as the liver and kidneys. And then T4, could be converted to T3 in the peripheral tissues. Make sure you also have an understanding of how these two hormones are created. We talked about thyroglobulin and the use of iodide, and how we created this fat-soluble hormone via exocytosis and endocytosis, and then how we release that into the blood across the cell membrane. Make sure you have an understanding of that pathway. Yeah, and I'm not gonna go over this in too much detail because it's pretty straightforward, and we talked about the hypothalamus. We spoke about the portal circulation. Make sure you understand what the portal circulation is, and you're able to identify where it is, right? This connection between capillaries from the hypothalamus going to the anterior pituitary. The posterior pituitary is not shown here, but we also spoke about the posterior pituitary and the two hormones that are released there, ADH and oxytocin. So make sure you're remembering that in your studies as well. And then the last thing we spoke about with regard to the endocrine system is understanding the fasted versus fed state, the absorptive versus post-absorptive states, what hormones were involved in each of those, right? So insulin is going to be associated with the absorptive state, glucagon associated with the post-absorptive state, and make sure you know the general function of what's happening between these two divisions. Between these two divisions. Okay, so I'm going to see a few questions here. Okay, so excellent. Someone is reminding me about the quiz questions that a lot of people got wrong. And then someone was saying, I don't see hormones unless you're still going over the receptors. So someone is pointing out that they didn't see hormones. I'm not sure where. If it's here in the endocrine system or if it's back in the autonomic. Oh, someone wasn't seeing the screen. Okay, that's going to be a problem. So can you guys see this screen? Oh, no. Okay, I see. All right, let me stop this.
Yeah, it's still stuck on the receptor screen.
Oh, thank you for pointing that out. That, okay, thank you for pointing that out. I don't think I shared this correctly, so it's probably still on that other screen. All right, okay. Can we see that now? Hopefully, yes. Okay, awesome. So this is the image that I was explaining in detail, and I'm sorry you guys couldn't see that, but this is something we drew in class, this hypothalamic anterior-pituitary axis. So you should have this. Hopefully you were able to follow along But if you weren't, here is what it looks like. And again, it's really straightforward, but I was just pointing out important parts of this that you really want to focus in on. So knowing what's happening in terms of the general access, understanding this particular access with the thyroid hormone, the short loop feedback, and then the long loop feedback. We drew this out in a little bit more detail. Also, make sure you understand the posterior pituitary hormones, which are not shown here, but we did speak about them and a little bit about their function, okay? So let's see if I see any other questions here. Okay, so what I'll do now is I'm gonna go to a few of the questions that were commonly incorrect over a few of our quizzes. So hang on, let me pull this up. In the meantime, while I'm pulling this up, I just want to mention it now that it's kind of fresh in my mind. I see quite a few of you have signed up for ANP2, which will be in the summer. Just a heads up, we haven't gotten official word, but we are strongly leaning towards conducting that online. In fact, you can kind of take that as being official from me, because that's the way myself and Dr. Schlater have been planning. So we're going to be team teaching or co-teaching in the summer. And so we're going to try to move everything online, just because that's the heads up that we're getting from the administrative side, so that when that announcement goes live, we will hopefully be already prepared accordingly. So just giving you guys a bit of heads up in terms of that. That means that labs will continue to be in line, kind of very similar to what we're doing now. Probably a little bit more integration of some other resources. There are some virtual resources that I've been looking into. So we'll probably try to expand the video quality and give you guys some different resources that are helpful in the absence of actually using human cadavers. And then phys labs will probably also be online as well. Okay. Okay, hang on, let me organize this a little bit. Okay, here we go. Oh, sorry, I also got a question here about fall classes. Fall classes are really up in the air. They're asking us to prepare either way. And so really there's no concrete answer that I can give you, but just know that we're preparing either way. So it's very likely that that may also be online as well. And not just, yeah, so we may see fall classes very, very likely be online. But again, that official word hasn't come out. They're thinking by July, they'll have an official word for the campus. But so far, that may be a possibility. So stay tuned on that one. Okay, medium. All right, so I'm going to go through some of the questions here. Actually, let me go back here and share this. Here we go. Okay. So hopefully you guys can see this, and this is the way I kind of tease questions out by whether or not they were marked as difficult or easy. And so I'll go over some of the ones that were that didn't perform as well as the others. So this is one. So parasympathetic postganglionic neurons release the neurotransmitter dash. Sympathetic postganglionic neurons release the neurotransmitter dash. So I think we looked at a clicker question that was similar to this. So really the difference here is understanding the fact that we're speaking about postganglionic neurons here. On the parasympathetic side, it's going to be acetylcholine. On the sympathetic side, it's going to be norepinephrine. And we could have switched this question around to ask about the preganglionic, and I think we looked at a clicker question as well to that effect, and that would have been acetylcholine for both. So I don't know if maybe students were mixing up those two questions, because we did have another question that asked about the preganglionic neurons, and the answer for that would have been both releasing acetylcholine, right? Remember, all preganglionic neurons release acetylcholine. But because this specific question is asking about the postganglionic neurons, here we see two different neurotransmitters depending on where we are. In the sympathetic side, it's acetylcholine. In the parasympathetic, in the sympathetic side, it's norepinephrine. In the parasympathetic side, it is acetylcholine, okay? So there may have been a bit of confusion in regard to that one. Let's see. Okay, so here, this was the second quiz, and we had 3 questions. So the first one here, how does latroxin, the venom of the black widow spider, cause muscle spasm and rigidity? And we spoke about 3 different toxins: quotoxin, latroxin, and curare. We described latroxin as stimulating acetylcholine release, and so that was going to cause increased acetylcholine at the neuromuscular junction, which would increase that contraction, that end plate potential on the muscle side, which will essentially cause muscle spasms, right? More neurotransmitter flooding the system, flooding the junction, which will increase the contraction, which would cause muscle spasms and then consequently rigidity. Alrighty. Let's look at some of the other options here. So it blocks nicotinic cholinergic receptors. This was what we described curare to do. So this response would have been correct if we asked about curare. And then this response, it inhibits acetylcholine release, would have been quotoxin. So the venom from the rattlesnake we described as inhibiting acetylcholine release, which would cause weakness and paralysis. So would curare. Both of these would cause the same types of symptoms, but they would be caused by different mechanisms. One is going to block the receptor altogether. One is going to decrease the amount of neurotransmitter. This one was quotoxin, and this one was curare. Okay, so make sure you also are able to distinguish those 3 toxins. Let's go back here. This other question that didn't do too well, which of the following is true of the absorptive state? So the absorptive state, energy input exceeds energy output. That means we're going to be storing up energy in our macromolecules. Think about processes like storing up, not gluconeogenesis. That is going to be the opposite. So processes of building hormones, building proteins, building fats, storing triglycerides, storing glycogen away into muscles, using what we need for the time, and then sort of putting the rest away for later. So that was in alignment with the absorptive state or the fed state. And the hormone here would have been insulin, okay? And then we can look at the other option that may have been correct if we were asking about the post-absorptive state. And that would have been, okay, so this doesn't make sense, right? If input is exceeding output, we're not breaking down macromolecules. So make sure you're also looking at what the response is. So this is not a correct statement. Input is less than output, and that's okay. So A or this option would have been correct if we were speaking about the post-absorptive state. So energy input is less than energy output, and so we're releasing or breaking down macromolecules to liberate energy. Okay, so the absorptive state would have been B, and then the post-absorptive state, which would have been in control by glucagon, is going to be option A. Okay. And then another question here that we didn't perform well. The metabolic adjustments that occur in the body during the switch from the post-absorptive to the absorptive state are largely triggered by increases in what hormone? And again, that would have been insulin. So if we're going to the absorptive state, that is going from glucagon to insulin. Okay, that one is pretty straightforward. And then let's see what we had from this most recent quiz, which would have been quiz 9. Okay, so we have 2 questions here. One question asked about the outer boundaries of the sarcomere. And I think I understand why a few people got this question wrong, or I think I understand. Okay, so the actual breakdown is not in alignment with that. So maybe that's not why. But there could have been confusion here. So the boundaries of the sarcomere are created by the Z disc. And so when we speak about a sarcomere, we're talking about from one Z disc to the other Z disc. Now, if you were reading into this a bit too much, you may have chosen the thin filaments because the Z discs are actually made by the thin filaments. So I went ahead and adjusted the grading on this question just because there may have been confusion here, but the accurate answer, the correct answer, the best answer here would have been the Z discs, okay? So from Z disc to Z disc is what we describe as a single sarcomere. And although the Z disc are created by thin filaments or actin filaments, it's really from that Z line to another Z line, and not so much from thin filament to thin filament, okay? I want you to have an understanding of that. And then a lot of people said thick filament, which would have not been correct, right? The Z discs aren't made of thick filaments. In fact, thick filaments are going to be in the center of the sarcomere. So make sure that you go over that because quite a few people selected the thick filament, okay? But if you selected Z disc or thin filament, that would have been correct. And then the second question here, which of the following physiological responses is associated with an elevation in the parasympathetic nervous system? And this would have been enhanced absorption, right? Remember, the parasympathetic is in control of resting, digesting, and glandular secretions, except sweat glands. And that is in alignment with absorption of nutrients or the absorptive state, okay?